Fasten your seatbelts, younglings. We're gonna learn you a thing or two about bacterial transformation. In the late 1970s, we first had success with inserting a human gene into the plasmids of bacteria. We now use this method to produce more pure and less expensive human insulin and human growth hormone. This is done using bacterial transformation, which is a type of horizontal gene transfer. Basically, the bacteria take up foreign DNA from the environment. Transformation was discovered by Frederick Griffith in 1928. He was studying Streptococcus pneumoniae. His studies led to him injecting mice with two different strains of the bacteria, and he discovered that one of the strains killed the mice and one did not. When he injected a killed form of the deadly strain, nothing happened and the mice remained healthy. However, when Griffith injected the killed bacteria and live non-deadly bacteria, the mice died. He had inadvertently discovered that DNA could be a transforming principle. In this lab, we will transform E. coli using a plasmid. The plasmid contains the gene for the green fluorescent protein and the gene for antibiotic resistance. The bacteria plated on the agar containing ampicillin will only survive if it takes up the plasmid. Without the plasmid, the bacteria will not survive. The coli that has the antibiotic resistance should glow green. The bacteria should glow under UV light. Now that we have this information, let's begin. First, mark two sterile 15 ml test tubes plus plasmid and minus plasmid. Next, transfer 250 microliters of ice-cold calcium chloride to each tube. Then chill both tubes in ice water. Once that is done, transfer E. coli from the incubation plate to the plus plasmid tube using a sterile inoculating loop. Suspend the cells in the calcium chloride until homologous by pipetting in and out into the fluid. Return the plus plasmid tube to ice. Repeat steps 4 and 5 for the minus plasmid tube with new sterile tools. Transfer 10 microliters plasmid DNA to the plus plasmid using an inoculating loop. Dip the loop into the plasmid and it will form a bubble within the loop. Spin the loop within the plus plasmid tube to mix the DNA with the cells. Incubate both tubes on ice for 15 more minutes. While waiting, you can label the petri dishes like so. LB stands for Loria broth, a substance that promotes cell growth. AMP stands for ampicillin. Heat shock the cells by transferring the cells directly from ice to a 42 degrees Celsius hot water bath for 90 seconds with constant agitation. Return the tubes to ice for at least 1 minute. Suspend 250 microliters of Luria broth to each tube and keep the tubes at room temperature.
Rest.
Remove half the cells from the plus plasmid tube to each of the plus plasmid plates. To do this, use a clamshell technique to distribute the cells into each plate. Roll glass beads within the plates with a back and forth motion to evenly coat the agar. Let the plates rest several minutes after taking out on the beads. Repeat for the minus plasmid tube.
This video is sponsored by Divine Labs for all of your experimental needs.
For more information, go to www.whydiditakethisclass.org.
Wrap the plate cover to the plate with tape and place the plate's agar side up in the incubator. The results from our bacterial plates were unexpected from what was intended. There was no bacterial growth on the LB AMP plus and LB AMP minus.
They ask you how you are, you just have to say that you're fine when you're not really fine, but you just can't get into it because they would never understand.
Long story short, our experiment didn't work. This indicates that the plasmid that promoted antibiotic resistance was not successfully transferred into the bacteria. It was also shown in the LB plus plasmid plate. There was bacterial growth on the LB plus and LB minus plate, but there was no green indicator to show that the plasmid was accepted. This could be due to many different factors that could prevent the bacteria from becoming competent. If the heat bath was too hot or cold, the bacterial membrane would not be able to become competent and would be unable to accept the plasmid. Bacterial handling could have reduced the alive bacteria as well. When handling the suspension, bubbles formed which could have harmed the fragile bacteria.